Are you seeing people coming in now?
Yep. Okay. Welcome to our event. We are ready to get started. We'll just give everyone a few moments to come in, just for those people who are running a little bit late. Sadly, Julia, my colleague couldn't make it to the webinar this evening, so I am running this solo. So please bear with us because sometimes we need to action some things behind the scenes. We'll just do some housekeeping while we're waiting. You can have your video on or off. I have actually muted all the attendees. So when we have questions at the end of the presentation, this setting will be removed. And this webinar is being recorded, and you should have been notified of that as you entered the webinar. If you have any questions during the presentation, please use the chat to enter them. You will also have a chance to ask them yourself at the end of the presentation. Please wait until we open up the question session. And if you want to speak, you will need to raise your hand by clicking on the hand symbol so that we can be fair to everyone. Once you have asked your question, make sure you lower your hand by clicking the hand symbol again. Okay, so what I'd like to do now is I'd like to actually introduce Paul. It is an honor to introduce a new friend of mine, Paul Sheppard, who hosts the Natural and Regenerative Beekeeping Facebook group. Paul originally comes from Trinidad in the Caribbean, or Caribbean, depending on how you like to pronounce that. It's just off the coast of Venezuela, and it is here that he started beekeeping in 1984. A move to California and then North Carolina in 2010 changed the way Paul kept bees forever, and he found a passion for keeping bees with a natural and regenerative philosophy and approach. His life journey took him to Portugal and now France, where he currently resides in a village that considers pollinators and does not use agricultural pesticides. We are grateful that Paul is a group expert in Be Natural Beekeepers Varroa Warriors and is able to present this webinar. Please welcome Paul.
Hi, everyone. Can you hear me, Shelby?
Yes, we can, Paul.
Great. Hi, everybody, and good evening, Australia. This is pretty exciting for me. Um, well, I'll get right into it because we have a lot of ground to cover. Uh, the first thing that I'd like to say is that regenerative beekeeping is really what we're looking at. I don't seem to have a control over my slides.
Just a moment then, Paul.
Okay,
You
here we
right?
go.
Yep, it moved on
Yeah,
there.
yeah. I had to unstick something. So there really is no such thing as natural beekeeping because once we try to put bees in a box or a hive and pull frames out and check bees and cut honey out, we're not really natural anymore. So what I like to say is what we're doing is regenerative management, where we are trying to get the bees to regenerate the traits and the abilities that they have to keep themselves healthy. And we're using methods that are closer to the natural ways that bees live. And it'll be something like, like the way they live in trees. And um, I started beekeeping in 1984, as Shelby told you. That is a picture of me there in a forest in Trinidad. Um, I and you can see just down in the corner there, I have a little map with a microscopic view of Trinidad compared to Australia. It's like a dot. So I started beekeeping in Trinidad in 1984. We started off with European bees. They were nice and gentle. And then in 2 years, we suddenly had Africanized bees. And wow, those were an experience. Very aggressive. And I didn't know really how, I was just a beginner beekeeper. I didn't really know how to manage them as such. So I would get stung when I was at home sleeping and just dreaming about them. They were quite aggressive. I our boxes got taken over by Africanized bees, and I opened it up one day expecting Italian, and I had to run for my life and dive in a pond. But that's another story. Um, I moved from Trinidad to United States where I did personal beekeeping in California. It was really difficult to raise bees there naturally. The bees were they were descendants and swarms from bees that had gone to the almonds, and they were pretty beat up, so they were difficult to deal with. I moved to North Carolina, in the United States there. They've just been hit by a horrible floods and disasters from Hurricane Helen. But most of these things are still there. I was a volunteer with the Center for Honeybee Research, which is still there in North Carolina, and worked with this guy in the middle here, Carl Chesick. And they had a whole research project on collecting data on keeping bees naturally and keeping bees in conventional ways. In other words, treating and all that sort of stuff. I am strongly connected to the Caribbean, and and really landed a fabulous opportunity where I was asked by the St. Lucia Social Development Fund to teach about 40 farmers how to do beekeeping. It was a fantastic project. For the first time, we got bees, a bee breeder and a bee supplier and a local guy to build boxes. And really, it was a wonderful experience for me. I also got a second opportunity to teach and work with the farmers with disabilities in St. Lucia. And we included a research project. So these are the farmers here on the left. And this is some of the data from the research project. The project only lasted a year. There were situations that were unavoidable. Number one being is that you cannot teach poor people about collecting data and research when they need to get honey and products to sell from beekeeping. And very, very quickly, this little graph over a year, the red represents the conventional hives. We worked this whole research project um, based on the same principles of the Center for Honeybee Research in North Carolina. So we were gathering data, weighing hives, checking, the strength of the different hives. In the back, the green is the untreated, unmanaged hives that were kept naturally. And over a year, they functioned much better. You can see that just looking at this chart, that the hives on the ends, because they were put in rows next to each other, the hives on the ends caught all the drifting bees and built up. So that is something that happened. As Shelby told you, I am now living in France where I help people uh, learn about natural beekeeping, and it has been a very interesting situation. The honeybees in France, they got hit with varroa and they lost a lot of hives and they lost a lot of feral colonies. But the bees seem to have rebounded, and now we have. I find my bees, the bees that I have. Let me put it this way: I bought in 2019. I bought 4 hives for myself from a local breeder. They were Buckfast Carniolan. And in the first winter, 3 hives died out. So the surviving hive is the one that I used to replenish my apiary. And I also allowed it to swarm and catch the swarms. And from that, now I have built back up. I can't put more than 7 hives in my yard because I'm next to neighbors. But I have 7 hives here, and I also help people manage their hives in a natural way. And we're getting a very good response from the bees. The bees appear to be regressing, or I don't know how you'd say it, but they're becoming darker. And we suspect that they are Apis mellifera mellifera. And they're having a lot, they seem to come with uh, varroa sensitive hygienic behavior. And I don't have any mites in, well, I don't say any, there are some. I don't bother to check because why bother to check? They keep surviving, they swarm every year, and they produce honey. So and this year I started extracting honey from some, but leaving honey for them as well. So here we come to what we are really about here today. And modern agriculture really is harming our planet. I'm sorry to produce these pictures to show you, but the batch rearing of animals and even crops, monocropping, all of these things are affecting our environment and also affecting the animals that we look after. Because in order to keep them in these environments, they get sick, they have to use a lot of pesticides and what's it? Forgotten for a second there. But they use a lot of pesticides to keep them alive. And that gets into the meats and into the animals. We're doing the same thing with the bees. Some people may not like this. They may think, but that's a good way to raise bees. I don't agree. I'm quite happy to say so. On the left, you see the type of production that we have to create queens. This has weakened the force of the bees. And things like on the right here, we see migratory beekeeping. Now, I know it's necessary. I know that things like the almonds, and also I think that you have almonds in Australia and you have to transport a lot of bees there as well. But these approaches are things that are definitely harming the bees. We have learned to graft and artificially inseminate and create, you know, all sorts of methods for increasing honeybee production and putting a lot of pressure on queens. I have a friend who lives in Hawaii where they actually breed bees for North America and Canada, and they use those queens to replace the queens that they have that they take on these migratory routes. In America, it would be like going up to California. They come from Florida, they go to California, then they go up north, and the same bees are pollinating almonds and strawberries and watermelons, and eventually they come back to Florida and they're pretty wiped out. And if we keep using this type of method of working with our bees, we're going to have problems with them. So you know about, I think you might know about Rudolf Steiner. He's the big biodynamic guy. And he said that in 100 years, we'd probably have problems with our bees. And exactly 1923, to now is 100 years plus one. So we are definitely having problems. Well, some people, some people are beginning to realize, and that's why I'm really glad that you're here to hear this talk. I am one of many people saying what you're going to hear today. I'm not the only one. Honeybees are the canary in the mine shaft. They they have collapsed, or at least they did. In the United States, the colony collapse disorder made everybody like wake up, like what's happening? And now we are turning around and we're starting to look at all the reasons what is causing these collapses. We have to prop up our bees with pesticides and uh, miticides and all sorts of everything we have to use to try and support the bees because they no longer seem to be able to stand on their own. Like before, years ago, bees, nobody bothered about natural beekeeping. Beekeeping was natural. I guess you in Australia really, that's how you kept your bees. It's not a problem. You just have bees and you take honey. But now the introduction of things like varroa and all the pests that are coming in and all the diseases like the deformed wing virus and all these sort of things are the straw that broke the camel's back. It's exposing what we have done with the bees as being not good for the species. Here are just a list of some of the factors that are causing the decline of the honeybees. Climate changes, can't get away from that one. Pesticide use in agriculture, and also the ones that we use in the hives. I'll talk more about that. Habitat loss, there's a big one. We're losing a lot of valuable nectar producing plants and systems. Improper apiary management. Some people may not like to hear that one, but I'll explain that much more. Pests and pathogens. Yes, we have pests and pathogens, but what's really causing the problem is that we have weak bees that cannot deal with pests and pathogens. And we'll talk more about that also. Competition among introduced species and native bees. There's only so much available in the system. There's only so much available in the area that can supply the bees with nectar and pollen. And if everybody's competing, somebody is going to feel the pinch. And what's happening a lot is that honeybees are actually outperforming the native species and taking over. And that's not good for the ecosystem. For nutrition, all sorts of crops that are sprayed with all kinds of pesticides, also using a lot of sugar as a substitute for honey, and high fructose corn syrups. We'll talk about that. So let's go on. So why are we talking about regenerative beekeeping? Why is that something that's worth thinking about? The use of chemicals to control pests and disease in honeybees is affecting them big time. I'll explain. Batch rearing of honeybee queens, you saw a picture earlier there of what it looks like. Drone suppression, we We have heard this since we started beekeeping. Drones are not good. You know, they drink all the honey. Swarm suppression, big problem. Big problem. That is one of the things that we have really decimated our honeybees by doing, stopping them swarming, or at least not, or using time splits or false swarms. Breeding, particularly just breeding for docile and productive bees and neglecting the natural process of things and beekeeper management practices. This is a crazy one. The last one there, people say, but I learned all of this. And um, what we find, because I am one of the founders of the Natural and Regenerative Beekeeping group on Facebook that now has 37,000 members. I'm not sure how many are actually members or who is a robot, but we have quite a few people listening. And one of the big things that keeps coming into me is that new beekeepers keep coming in and saying, okay, well, I just bought bees. What do I do next? Oh, this is happening. What do I do now? The way that we are learning beekeeping is affecting the bees because everybody comes in and they keep opening hives and checking this and checking that and, using all sorts of techniques to increase production. So I don't know if you ever seen this slide, which is Apis henshawi, named after the guy who discovered it, Henshaw. It is a honeybee in amber resin, and it's a fossil for dating back 33 to 23 million years. So I'm being conservative. Honeybees have been definitely around for 33 million years or 23 million years. And other experts will tell you way back, further back than that. So they've been around and using techniques to stay alive. And they have managed to come up from Indochina, from the Borneo side, come all the way across India, apparently down in Africa. I'm not actually sure if there wasn't another species from Africa coming up, but they came to Europe. They encountered the glaciers in the ice ages, and as those glaciers backed off, they followed them up. And Europe has the Apis mellifera. You have the African bees, the scutellatas, and all that sort of stuff. So honeybees have managed on their own for millions of years. And how long have we been beekeeping? And look at what's happening. Isn't something kind of off? So I talk to this guy, Tom Seeley, from time to time, and he is a great guy. He's written many books. Yeah, you must have heard of Honeybee Democracy. And Tom Seeley has been one of the foremost pioneers on the natural ability of honeybees to heal themselves and regenerate and deal with varroa without crashing. And basically, that's the talk today, what we're going to be talking about. So he had a project in the Arno Forest in New York for the university that he was with. They had done a census in 1978 of the bees that were living there. And after varroa came in 2002, they went back and they checked for 3 years the population of honeybees. And the last paragraph here says that they went back out of 9 colonies found in the whole forest. They found 8 living on the western side of the 2/3 of the forest. So they didn't do the whole forest. And they found bees surviving. I remember when I read this years ago, and it hit me like a meteor. It just came and boom. turned me on to what are we doing? Honeybees have a natural ability to take care of themselves. This is a picture that was taken in France a year or two ago. And this is Vincent. And now, Vincent, if you're listening to this, I'm sorry if I pronounce your name wrong. It's Alboy or Alboy. He is an entomologist and a very interested in the natural bee populations in France. And not too far from here, I have a friend who works with Vincent, collecting, monitoring wild swarms. There are several. I mean, he took me and showed me maybe 30 living in walls, living in tree trunks, living in the roots of tree trunks, living in chateaus. One chateau had 15, I believe it was 15 colonies living in the walls. And for years, some of them over 8, 10 years. This picture here is very interesting. I use it a lot whenever I'm talking about natural colonies. So this is a colony that moved into the window of a French farm, and the farmer just left it. So, in France here, we have these shutters and then we have the window. And they got into that cavity and they built their colony. This colony is about 20 years old. And it has existed for all that time through varroa, through everything. In the spring, it would expand and get bigger and produce honey. And then in the winter, it contracts and it gets smaller, and the wax moth come in and they eat up the extra comb. And he is pointing there at a colony of hornets. I think they're European hornets, but they are parasitic to honeybees. But they're living in a harmonious relationship. There's enough hive and honey for the bees, and there's enough bees for the hornets. And they just live together in harmony. It's a very interesting situation. I went to see the hive, but it had finally perished. And probably due to a nearby pesticide application. But isn't that an interesting thing to look at? It is very possible for bees to live on their own without interventions, deal with varroa, and be in a natural way. I know some of you are here to hear about this, and it's the Cuban experience. So let's give you an idea. So you know where Cuba is, and Cuba is kind of cut off from almost the rest of the world, definitely cut off from the United States, they have imposed an embargo on Cuba. And when varroa came in, I believe it's 1996, when varroa came in, so they couldn't get what was available at the time, the AP stands and all that sort of stuff. So they made a national decision to not treat. And it was, of course, quite devastating to the entire beekeeping industry and to the bees in Cuba. They lost feral bees, they lost hives. They said, I saw a report that said 8,000 hives were lost. Now, they had over 100,000 hives at the time. And I find this a very small loss. And I was wondering why that happened. Right? The bees in Cuba are European bees. They are Apis mellifera, and ligustica, and maybe something else, but they're not scutellata. They're not connected to the Africanized bees, which populate all the other Caribbean islands, like even Trinidad coming all the way up. They don't really populate. They don't get to Cuba. So this is an example of an entire country I think Cuba is something like 45,000 square miles, you know, around there. And this is an entire country that took a decision not to treat for mites. And guess what? In a few years, they bounced back. They started getting resistant colonies, and the colonies started showing these traits: removal of mites, um, reducing fertility of the varroa by capping and uncapping. All of these things were happening with these bees. Where did that come from? You know, a memory that just came back because of a stress. A pest arrived, and these bees in their natural environment were able to respond by waking up some old traits and perhaps learning some new ones. And now Cuba can boast, they have, what is this? This says here 220,000 colonies treatment free. And this was an old uh, excerpt that I took here. And look at the production. People say, oh, naturally kept bees don't produce as much honey. Well, look at the experience here. You know, I kind of wondered why it is that Cuba was able to actually uh, do this in one go, whereas other countries like United States and UK, Europe, why was it so hard to, it took 10 plus years for the bees to start changing in the feral colonies. But in Cuba, it happened much faster. And it just occurred to me that if it's done as a national effort, if everything happens, everybody goes natural, right? That means that there's nobody breeding bees that they're treating, which puts drones into the environment that do not have the resistant genetics, or have diluted resistant genetics. So that was a very interesting, that's a very interesting kind of balance. Have we, in the whole world, made the wrong type of decisions when it came to varroa coming in? We panic, we start treating, then we keep adding treatment bees treatment drones to the environment that mate with queens, and the whole development of the resistant trait takes much longer to come in. There are places all over the world, most of North Africa, many places in France. In Europe now, they're talking about resistant behavior, and there are over 2,000 natural beekeepers. There is anybody that's got Africanized bees, the Africanized bees have resistance. They don't get, they deal with varroa. It's not that they don't get varroa, but they can exist or tolerate varroa and control them. There are other places, the island of Dominica in the Caribbean, which is a very interesting case. I've spoken to the beekeepers there, and they have naturally resistant European bees in the middle of Africanized bees everywhere, and they don't treat their hives. So it's really interesting that all over the world, in parallel situations, honeybees can evolve or re-evolve and have these traits. I hope I'm not burning your ears, but this is one of my passions, and I can talk for a very long time. So, anyway, so what is honeybee resistance when it comes to varroa? Is it resistance or tolerance? All of these are questions that people are now looking at. The traits of varroa colonies for long-term survival is associated with recapping. So I think you might know what recapping is. It's the bees, the worker bees So there are a couple of different, labor assignments. So they're like sniffer bees that come along and sniff the hive and go like, there's varroa in there. So they uncap a pupae, white-eyed, even sometimes later with the pink eye, they uncap them. And then there's another batch of guys that come along and go like, this is an uncapped hive. We have to close that up. So they recap it. I heard yesterday that can sometimes happen 17 times. Isn't that interesting? That, what it does, is completely disrupts the varroa reproductive cycle. And you get reduced, reproduction. They also tear out the, the pupae and in some cases cannibalize them or throw them out. It's a trait that you see too in chalk brood, the throwing out of the mummies. Grooming is another aspect, but grooming only takes care of the bees that are outside, and which people try to stimulate with sugar shakes, but it's not the only trait. But the recapping trait is one of the most important ones to look for. And that is a whole lecture in itself. The other thing that the bees do is that they chew the mites. They bite off the front legs of the mites. And if you have a screen, not a screen, yes, a screen bottom board with a insert board below that, you can probably find bits and pieces of the varroa. You can find varroa with the legs missing. You can find the antennae of the pupae as the bees have, killed the pupae and pull them out. You see all of these things. There's a very good book by a guy called Paul Honigman that just came out. It's called the Observant Beekeeper, and it gives you all the things to look for when you're looking for resistant traits. So here we go. This is why we're here. Am I going on time? All right. Okay. So the following ideas were suggestions and were contributed in part by myself and by members of the Natural and Regenerative Beekeeping Facebook. It's also taken from many research projects and papers. So the biggest thing is, if you don't mind me taking a drink here and wetting my throat. We have decided, or when varroa first hit, it was a huge. Of course, the media got involved. It was panic, freak out. Everybody's freaking out. Everything is gonna die. How are we gonna save the bees, right? Well, we have to come up with chemicals because what do we always do? We see a problem and there's a chemical for that, and of course, these chemical companies will step in. I won't mention names. But the first thing everyone decides is that we have to treat. So they decide that they want to use, the first one was Comophos. I think Comophos is an organophosphate. Deadly. They were using an organophosphate on an insect to kill an insect, right? And there's a guy here in France called Dr. John, what's his name? Oh gosh, I've forgotten his name there for a minute. Anyway, he started treating with the Comophos, and he couldn't handle the odor of the Comophos. It was affecting him personally, and he felt, why am I treating my bees with this harsh thing? So he decided to go natural. So he stopped treating, and he says he lost 2/3 of his bees. you know. Right away, varroa took it over, but then he got survivors. And then he took those survivors and he made new colonies from the survivor. A simple, everybody can do it. You don't have to be a scientist. You don't have to have a PhD. All you have to do is have strong will and a strong back. And what you do is you start developing your hives, your new hives from survivor colonies. And this is how you start breeding your bees for mite resistance. After a while, he said that he couldn't find mites in his hives, and he needed mites to create the response. So in France, he advertised in the paper for mites. He needed mites, and of course the French were like, "Ugh, Monsieur, you're crazy," you know. And he was actually looking to get mites to introduce into his hives to create the resistance response. So that is an interesting thing. Typhus is his name. Sorry, my memory sometimes leaves me. The other thing about treating is that when you start using all these pesticides, first of all, what you're doing is that you're selecting for mites because you're reducing the resilience of the bees. And the mites, yes, some will die. If we know about insect resistance, some die and some live. The ones that live are stronger, and they create a new population from the stronger mites. So this is what they've been finding is that the mites were getting stronger. The mites were building up resistance. So they changed the chemical. They started to use fluvalinate, and now they're using amitraz. And of course, there's all kinds of problems with these synthetic pesticides. So now people are going more like they're saying it's organic beekeeping. So we are going to use um, things like oxalic acids and formic acids and all these sort of things. But the truth of the matter is, yes, organic chemicals are better for using, but the most important thing is that if you use any type of treatment, you are delaying the resistance process. If you use anything to control mites, you are actually delaying your bees developing resistance. Now, I can hear a lot of you freaking out right now, but it's a way of doing it. And maybe it's not a full across the board type of thing. The other thing about chemicals, especially the synthetic chemicals, is that they become embedded in the wax. There are, you know, they have checked up to over 100 different chemicals embedded in honeybee comb and wax. Not all in the same place, but in different places, and depending on what area that you live in and whether people use pesticides. I am so lucky to have found a little village in France where they it's dedicated to honeybees. It's a pollinator oasis, and nobody is supposed to use pesticides. So my bees have thrived, I must say. And these are important things, to create little oasis or little pockets of natural natural environments, natural foraging. But the chemicals in the wax is a huge problem because we are still having that problem up to today. Wax is a lipid, a fat, and these chemicals get into it and they can't get them out. And the other thing is that these chemicals have synergistic effects or synergistic relationships with other chemicals in the environment. And nobody actually knows, right? They're trying to do research, but nobody actually knows what this chemical and that chemical with stuff like amitraz being put in a hive is going to do. And we're sitting here wondering, why are our bees failing? Why is it that our bees can't seem to, uh, to handle varroa or anything? Why is it that our bees are getting all these diseases? All of a sudden it just seems that bees are getting more and more and more problems. The other thing about chemicals, when you put them in, it disrupts the balance of microorganisms and beneficial insects. You you must have heard about pseudoscorpions, There's actually a group on Facebook dedicated to pseudoscorpions. These are little scorpion-like, but not actually scorpions, but looking insects that live in honeybee hives sometimes and eat varroa. They pick varroa off and they eat it. It's some people want to put 200 pseudoscorpions into your hive. That's not the way that you deal with this. And then you talk about the microorganisms, the beneficial um, flora and fauna, the microscopic balance in the hive, not only in the hive, but in the honeybee gut, which is very important. You're getting learning difficulties in honeybees due to glyphosate, because glyphosate is really a herbicide, and people say, oh, well, I even use herbicide around the hive to keep the grass down. But glyphosate gets into the honeybee gut and affects the microflora in the honeybee gut and causes all sorts of learning problems and difficulties, and affects longevity. So how do we actually go about this whole process? As I talked about earlier with John Cleefus, you start trying to raise resistant queens. Now you can't just take your whole apiary, and I'll actually give you pointers on starting to develop the resistant process without destroying your whole apiary, not going cold turkey and putting all your hives up for varroa to destroy. But once you start this process, you can use queens from survivor hives and requeen the hives that are not resistant to varroa. The queens have the genetic information in them, and if you make all the changes that I'm going to suggest, you can help them. So I have a beekeeping friend, and I'm hoping that he will actually talk with Shelby and Julia. He says when he walks around his apiary, he sometimes gets queen cells, and these are queen cells from resistant hives. And he actually will cut out a few, put them in his pocket, and walk around and requeen a couple of hives that are not doing so well. And this is a way of handling it. You have to use, the bees are using their natural instincts to develop the resistance, and they have it in them. And this doesn't take, as I said, a university degree. Anybody can do this. I don't believe in using artificially inseminated queens. They only use a few drones, seminal fluid, is it sperm from a few drones with traits that they want. And I spoke to Tom Seeley about this, and he said very often what they're doing is that they are selecting for two behaviors particularly. One is production, and the other one is being docile. And we are losing the other most important traits. Honeybees are genetic jugglers. They juggle. When they leave when the virgin queen leaves the hive and flies through a drone congregation area, it is just amazing how this natural selection is set up. The strongest drones with the biggest eyes, and they get the first chance to inseminate the queen, so that you're getting strong genetics passed on and passed on. But if we keep producing weakened hives with weakened drones, and they get into the fray and they get a chance to mate, then we are just passing on this non-resistant genetics. I don't believe in using artificially inseminated queens. Natural selection, natural mating is quite frankly the only way you're going to get this juggling. We need to leave it up to the bees. They have millions of years they've been here. They know much better than we do how to be bees. We think we know how to be bees because we have all these ideas. But really and truly, if you look at it, our ideas are kind of causing problems. You monitor your colonies for varroa sensitive hygienic behavior. There's a lot of ways you can do that. You look for things like, we call it bald brood. In the old days, you used to call it bald brood. It's like when you open your hive and you can see the brood in the cell, but there's no cap. That actually, and of course, when those things happen, people change their queens. But something is dancing around the screen here. I'm not sure what it is. Anyway, sorry, I lost track there. Oh yes, the brood, the ball brood. When you actually see that, it's a possible indication of varroa sensitive hygienic behavior. The bees are uncapping the cells and recapping them. So you have to set up a monitoring system. You check your hives for that. And you check the insert board at the bottom of your hive for the other things like the fragmented uh, bees and the fragmented varroa. Last thing on this list here, use local queens. Local queens are adapted to your microenvironment. For years, we have been bringing queens in from overseas. I'm going to mention this again in other things, but you're bringing queens from away and from even outside your area, and they are not adapted to. Shelby, something is drawing red marks all over the screen. Are you getting that? Okay. Anyway, I'll continue.
Yes.
Somebody's got something on, I think. Anyway, so yeah, really try to use local queens, queens that have been developed in your area. Everybody's the breeders are trying to create these VSH queens that have the magic bullet, the silver bullet that will take care of everything. But they take them out of that environment that they were raised in, and they put them in another environment, and they don't know. They're not in sync. They're out of rhythm with the environment that they have been put into. And bees really use rhythm as a way of, working with nature. Remember, they're tuned to flowers, they're tuned to the environment, they're tuned to the seasons. And I'll talk more about that. So are we going okay for time? It's almost an hour. So don't use imported queens and genetics. See, I say here the imported queens, they dilute the natural ability of the local queens of the local stock to resist disease. Breeders concentrate, as I said, on production and um, docile bees. And the thing is that these queens last a year, maybe 2 years for the most, and sometimes even 6 months, you have to replace your queens because of the way that they're produced. So I say, learn to set bait hives and catch swarms, especially your own swarms from your resistant hives. Be careful that if you're in an area where there are a lot of commercial beekeepers treating hives, you're going to have a hard time trying to create resistant bees in your apiary. And I make some suggestions later on. You see at the bottom here, you have in Tasmania, I hope I'm saying that correctly, Apis mellifera, quite a lot of that. Yeah. And I think I have the next slide, I talk about that. Yeah. Shelby sent me this. It's done by Nadine Chapman. It's very quickly, and I hope you can see it. These, the frame on the left is managed hives, and it shows the mixture of species in the colony, in the individual bees, the Western European bee, which is the Apis mellifera, the Eastern European bees, which more the Ligustica and the Carniolan's and that sort of stuff, and the African bees, which I assume is more like the Scutellata. And if you look, and on the right is feral colonies. Now I want you to do something. I want you to kind of like just go back a little bit and look at these two pictures, these two frames with, I'm sorry, is relaxed eyes. In other words, you're not really trying to look at anything in particular. But what do you see? Well, for me, the first thing that struck me was there's a lot more black dots on the right in the feral colonies than on the left, the managed colonies. So it tells me that the bees that are going into the wild, the feral colonies, are reverting to more genes of Apis mellifera. And we have found here in Europe that the darker bee, and there are societies for what they call the black bee, these bees seem to have a better adaptation to varroa and seem to have the traits for dealing with varroa. It's an interesting thing to look at it like this. And I think it's worth your scientists and your people trying to figure out how that works and how it is beneficial. Propolis is very important. People go into hives and you see them scraping out the propolis. Tobin Schiffer did some really good work on propolis and the propolis envelope inside the hive, how the bees create a propolis envelope. The propolis, it waterproofs the hive. They can use it to reduce and clog, patch up the entrances and all the leaks, like the air leaks. It has antimicrobial, antifungal, antioxidant properties. But here's one that on a paper that I just read. It has insecticidal properties against varroa. So the bees, the humidity in the hive condenses on the propolis and runs down. It is rich in whatever the properties that the propolis have, and the bees drink that or share that, and seem to have. It helps. It aids in their fight against varroa. So it's a very important thing not to take the propolis away from your hives and crack and break and scrape. Pollen, very important. People collect pollen for health purposes for humans. Everything is in a balance. We're all in a balance. You take pollen away from a hive, you throw the whole workforce off. You break the rhythm. And what has to happen now is that more bees have to be recruited to gather pollen, and less bees will go out foraging for nectar, and less bees will actually be doing housekeeping, chewing varroa, doing that sort of stuff. We break the rhythm, we break the balance, and the hive has to adjust immediately. Immediately it has to adjust. And then we wonder, well, why is this hive not performing? Why is it? people go into their hives. I talk about this a little further on, but people go into their hives way too often. Queen excluders, I really don't believe in them. I don't use them unless I want to hive a new swarm and I want to try and get the queen to stay. Never use them in honey production. Actually, not really that interested in honey. More interested in good bees. I don't know if any of you have ever noticed the bees will start propolizing excluders and screen boards, top screens and bottom screens, in an effort to control the air and the movement of air and the humidity in a hive. So I don't use these. I get my bees occupy a brood chamber. I just run a brood chamber and a super, and the super is always full of honey, and the bees really don't come up. Maybe just a little bit, in the middle section. The actual shape of a honeybee colony is kind of like, more like a circle. So imagine when you put a excluder above, what you turn it into this, which is kind of different, and you move the honey away from the brood, and the bees have to squeeze through. It's also called a honey reducer by some beekeepers, and I don't recommend using it. A lot of beekeepers sometimes they have their excluder in there and they trap their bees below that, and in the winter when the bees want to rise up with the honey, they can't.
Oh, we're at 7 p.m. now, just to let you know. So another half an hour to go.
Wow, this is going longer than I thought. I'll try to go through quickly. Um, don't use foundation, the store-bought foundation. And I think it's not so bad right now in Australia because you haven't really been treating bees, or maybe you have been treating for uh, what do you call that? Small hive beetles. But agricultural chemicals get into the foundation sheets, and I explained earlier how that affects the hives. Most foundation comes with a preset cell size. And this is one of the biggest problems because these cell sizes are what governs the size of the bees that come out of them. They've gone up to, I'll show you how they measure it. They've had this large cell size because somebody thought, oh, bigger bees will bring in more pollen, and then they went, oh, smaller cell sizes will reduce varroa. So now we have smaller cell sizes. It's all the different side of the same coin. Bees make their own cells, and one frame will have more than one different type of cell. The bees even put drone brood around their worker brood, so that as bait for varroa. Just heard that story and I thought, wow. I mean, the bees are like cowboys. They can corral varroa into one area that they are allowed to as a sacrifice. This is how the cells are set up and measured. They count 10 and they measure that distance and then they divide that by 10. So they started off with 5.4. It even went as high as 5.9 millimeters per cell size. So using these embossed foundations are not good. As in the previous slide, I showed you, I use just a strip. I use wires, but I use just a strip, and the bees build comb on that. Drones are very important. They are a huge, important part of your bees. they have the genetics for the resistance. Uh, there's so much to talk about here. You know, conventional beekeepers talk about the mite bombs. They talk about mite bombs that they say non-treatment beekeepers produce mite bombs and destroying their bees. Well, I think it's worse the other way around. The, the treatment beekeepers are producing drone bombs with non-resistant drones that are keeping the colonies down and keeping them from you know developing their resistance. I've opened a hive and Ferry has also seen this. We open the hive and it's like 3 or 4 frames of drones. And you're like, whoa, I've got a drone laying queen. But you're looking in a few more, but there's worker broods, and it's only at a certain time of the season. And then the colony reverts back. They start making worker brood again. The bees know that they need to 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 have drones. They are trying to develop drones. When you put um, foundation into a hive, it's really specifically for worker broods, not for drones. Don't suppress the drones. I'm kind of speeding up here to try to get to the question and answer section. Allow for swarming to take place or use time splits. Important. And very important for the swarming. In your initial experiences with varroa, swarming or time splits will create brood breaks and give you some breathing time. Leave honey for the bees. I think you must be doing that already. There's a beautiful picture of honey in St. Lucia. They use empty rum bottles, which I contributed quite a few, and with some nice honey. But honey has all the necessary ingredients, and it has a toxicological, sorry about that, relevance, especially in environments where bees are exposed to agrochemicals. We take that away, we start feeding them sugar, and sugar comes from highly industrialized systems. It's not I don't say don't feed, I just say don't feed to replace honey. Feed in extreme cases of starvation. The rhythm of the bees, I wanted to say a few words about this. Obviously, I don't think we're going to have time. Anyway, um, honeybees have been in sync with nature from very beginning of time. They have an internal clock. The foragers will actually wait inside the hive on the extremes of the colony, away from the dance floor. They will wait until such a time when the flowers are producing the most nectar that they fly out. They're economizing energy, they're economizing time. Everything is in a rhythm, and bees use rhythm a lot. We put they communicate with dance. They communicate by sending vibration through the hive. It's very important not to break those rhythms. Use single stands. Get away from the rows of bees. It creates drift. It helps um, spread pest and disease horizontally. Your individual stands are better. Alternative hives, skeps, worry, top bars. The one in the middle above the top bar is a hive I created from scrap wood, which is about 4 centimeters thick. Um, and then log hives. Um, Eliminate inspections into your brood chamber. Probably one of the hardest things for people to do, but one of the most beneficial. I have brood boxes I haven't been in for 5 years, and the bees are thriving. I wonder sometimes if I'm just trying to see how long they will actually take that before they leave that hive. Regular inspections disrupt the entire hive and break the rhythm of everything and the scent and the temperature. Remember that. You can tell a lot about entrance activity about the activity in a hive through what's going on in the entrance, and you can also find out a lot from your insert board. Keep at least one permanent unmanaged colony in your apiary. The one on the left there is a locally manufactured by a friend of mine, Costos Epium. That's not his name, but that's his company. And these do very well. There's a supply of untreated bees in these hives. Consider other pollinators. I hear that you guys have, very high levels of feral colonies in your in your bush. So you have to be careful that you're not overpopulating areas with apiaries that have like 40 to 100 hives. Keep hives off the ground. I think you know that. Just a very quick note, the top picture is me with one of my clients' hives. That area flooded and the water level came right up to the bottom of the super. The bees went up and survived the flood. It's very interesting, very good. And that stand is about 60 centimeters above ground. Use less smoke. I'm trying to do that, but let me tell you, if you hit a very hot hive, keep your smoker handy. I'm kind of trying to speed up. Collaborate with other beekeepers. Together we are one. We are strong. Divided, we are. If you got one guy here that's natural and one guy here that's conventional, you're going to hear all the arguments. Believe me, I've been through it for years. Plant more native gardens. It's a holistic approach to beekeeping. Try to plant as much native plants and flowers around your area. If you have, even if you have a windowsill, you try and put those out there. Changing your apiary to regenerative management. This is almost a lecture in itself. Amy, would you like me to take questions? Because I don't think we have much time.
We haven't had that much in the chat area, so you can continue quickly, Paul, and we'll probably have about 10, 15 minutes left for questions.
Okay, so this one is about changing your apiary to regenerative management. I advise that you don't go bond. Bond is like referring to James Bond, live and let die. Just leaving your bees. In your Australian situation, your bees are varroa naive, and probably you will lose a lot. If I would suggest that you set up a small apiary with one or two of your good colonies, a little bit away, not too far, because you don't want to be too far from the environment that you're in. I would suggest that you form groups and you do this, and you let these bees be natural, and you deal with them just the same way that we are dealing with all these suggestions that I gave. Monitor your apiary. Monitoring for varroa-sensitive traits is an important part of developing resistant honeybees. That's another lecture in itself. I have some links that will link you to some beekeepers talking about that. Now here's kind of what you might think is contradictory, but I think it's important in your Australian situation is to treat your initial apiary, the one that you take your test bees from. You treat it with low applications of reduced miticides such as thymol, but organic stuff, but reduced applications and solutions. And you do that just to keep your bees alive until such time as you can start requeening them. I don't know. Everybody is different. There may be some just backyard beekeepers, but there may be some guys that are into, um, they have like a 50 hives or so. We overall have to have a different approach to agriculture. Um, we are killing insects. Germany almost does not have insects anymore. And this is where we could end up. In China, they're hand pollinating trees. It's something that we just don't want to get into. This is a great thing by Gunther Hawk, the biodynamic beekeeper in North Carolina. I met him. More than we can imagine depends on the ability to turn a crisis into an opportunity for change. You're in a crisis, but you have an opportunity to change. The varroa is there to wake people up. This is my contact information, and I hand over to Shelby. Sorry I took so long.
Thank you, Paul. We will, and please don't apologize. It was all very interesting. So we will now open up the questions section. I've switched off the automatic muting so that you will be able to unmute yourselves now. Remember, if you want to ask a question, you'll need to raise your hand and we'll take them in the order that they come in. We'll also go through the chat quickly and look at those comments and questions first. So we do have some things in the chat already, and we'll start off with those. So I actually posted something first, Paul, and it was, and then we'll, excuse me, then we'll organize to hear from person number A, who we'd love you to identify yourself so that we know who you actually are, because you've got quite a few comments and we'd love to hear from you. So while you're getting yourself organized, I'll just jump into my question, which is you mentioned about the uncapping and the fact that some bees naturally do the uncapping and recapping, etc. Is that any way to assist us to help promote that trait by assisting the bees to do any uncapping, or is that not something that would help to promote that?
Well, I'm not sure what you mean by assisting the bees to help. I think what you're doing is that you're monitoring for it. You're monitoring to find that trait. When you see that trait, that's the colony that you want to promote. That's the colony you want to breed your bees from. You want to make splits from that, and that all helps. Remember, it's a whole holistic, everything is connected. It's not just one thing. Does that answer your question?
It does. So we need to let the bees do what they do best, but then monitor them to see if they're doing any of this uncapping or recapping, and that's the hive that we need to look at
Yes.
promoting. Yeah?
Yeah,
Okay.
exactly.
Fantastic. All right. So next, let's open it up the forum to person A. Can you identify yourself, please? And would you like to ask all of your comments or questions? Person A, do you want to take yourself off mute and comment or answer or ask any of your questions? Okay. Cynthia, you were next on the chat. Did you want to explore anything more with your question? Hello, Cynthia. Did you want to take yourself off mute?
Okay, can you hear me now?
We can.
Oh,
I
right.
can hear you.
Okay, Paul, I was talking to a French beekeeping friend last summer. I was in France, and he said that many French, in fact, you ended up answering this question, but I just wanted to see what you thought. He said that many French beekeepers take their brood box and put it straight on the grass. And there is this creature, this little bet that goes in and kills the mites, a kind of scorpion. Now, then you went on, I said, oh, you're just talking about that right now, so you can ignore my question.
know how to do it, then.
But you weren't terribly keen on that, I noticed.
Putting the hive on the ground?
Well, no, it was the scorpion, the little scorpion spider or scorpion something or other you were talking about.
The
Pseudoscorpion.
Pseudoscorpion.
Yeah. No, don't get me wrong. I'm in favor of it, but I'm not in favor of having that as the only approach.
Okay.
You know, a natural hive you will allow all the beneficial insects and the harmony to take place. You know, pseudoscorpions, you name it, propolis, blah, blah, blah. You know, but I've never heard of the guys actually putting their hives on the ground to get the pseudoscorpion. You know?
But in fact, we don't have that insect here, so it's not going to work in Australia anyway. But the other methods, of course, we can use.
Right. But maybe there's something that you do have that works with the bees. I don't know if anybody's researching that.
No, I don't. Well, I haven't heard anything, but maybe there is somebody out there. Thank you. Anyway, it was very interesting, Paul. Thank you.
Thank you.
Thank you, Cynthia. We have, is it Booby or Bubby? Did you want to ask your question?
Hear me?
Oh, yes. Just say,
good.
speak up a
Thank you
little
very
bit.
much. Thank you. Yes, my question is this: has there been any field study, and this is, say, in Australia or in Europe, using native, within Australia, the native bee, stingless bees, or in Europe, anything that would be or called as native to the land? Has there been any field study of them to gauge resistance to varroa?
So your stingless bees, I think, are pretty different to Apis mellifera. I can't remember the identification name of stingless bees, but if we're talking about the same thing, they're kind of black, sticky, right? So I don't know about that. I honestly can tell you, I don't know if the stingless bees are resistant.
Okay, thank you.
Okay, sorry, can't answer that one.
That's okay, Paul. You can't be an expert on everything in Australia as well, so thank you. Just to answer that question, there is some information to say that the native bees are more resistant to varroa, and there's been no information regarding native bees being impacted by varroa at this stage. I don't know whether that will change over time. Okay.
We hope the earlier the better.
Exactly. Matcha. Oh, Macca. I'm sorry, Macca. We'll organise to post those slides from Paul in Be Natural Beekeepers Varroa Warriors Facebook group. So, and the recording will also go there as well as out to everyone. Kay, you're next. You have a question?
Oh, hi. Thanks, guys. It's been really interesting. I've got a couple of questions, actually. So rethinking now to have one hive that just leave natural and not do anything with it, just let it go. And I think that would be an interesting task to take. But how do you go? So where I am, I've got varroa in my hives, but they're below what they call treatment levels. So I'm just getting like one varroa in my tests. But we've got a lot of American foul brood in our area. So I've had one hive that was affected by that. So if we leave them, how are we testing or how are we checking for that? Because in New South Wales, we have the orders are that we have to conduct a full inspection every twice a year.
Yes.
So we can't go against those orders. So I guess that's my question is, if you're leaving them alone, how do you monitor for any other pests or diseases?
Okay,
Good
so that's a
question,
really,
Kate.
yeah, that's a really good question. So the thing is that twice a year is basically what I'm getting an echo, but anyway, twice a year is pretty much how much I check inside my hive. That's no problem. What I don't know how you have your hives configured right now, but I would be trying to get this hive to swarm by not putting extra boxes on top of it. I think you're in your spring, or you're coming into your spring, and you're going to start to get a flow shortly. I would try for this hive to swarm. That's good. Go ahead. Say, you can say what you want to say.
That wasn't me, but I just muted because you were getting feedback. We have to destroy those hives here completely. We've got to burn everything or have them irradiated. So, but I've done that. Like I don't have it at the moment, but it's just that that's one thing that I'm checking regularly for is AFB in my hives. So if I'm not doing regular inspections, that just worries me. If you, I'm actually inspecting every 10 days and I'm testing for varroa every 10 days because that's what has been recommended by our Department of Primary Industries in New South Wales.
Right, I understand your situation. It's pretty much a catch-22, you know. Damned if you do, damned if you don't. It's kind of
I'd be more happy not to go in as regularly because just disturbing what you're saying, the rhythm. So
yeah. You know, one thing that I have found with natural beekeeping and natural hives, and I can tell you we see a lot of it, is that there is no American foul brood. There is no European foul brood. There is no deformed wing virus. Once the balance is restored and the bees are healthy, these things don't exist as much. To be honest, don't think I have ever even seen American foul brood in all the years that I've been working with bees. It just don't seem to come into natural hives. You know, going in every 10 days is kind of going against the grain, but I understand that it's being demanded of you by your government. That's kind of this is really a dilemma, because disturbing the hives that often really breaks up the balance. Over to you.
Thank you, Paul, for that. Because we're close to time, we do have a couple of extra comments and we may go about 5 minutes over. So my apologies for that this evening. But Eric, did you have anything that you wanted to contribute at all regarding your comment? If you did, this is your opportunity to take yourself off mute now. And then we'll go to Ronnie next.
Yeah, I think this, can you hear me?
Yes, we can.
Yeah.
It's just with the ordinary Langstroth hives and the book scorpions. There was just from Torben Fischer as well some experimentation. They just don't thrive in normal Langstroth hives because they don't have places to hide and propagate, and it's too wet for them in there. So that's not really going to be helpful in commercial beekeeping. and we have them in Tasmania, like Ronnie said as well below. And just maybe suggestion from like I don't know if they're there or if they can unmute or something, but if you just read out the questions and then Paul could answer them.
Sure. Okay.
Cool.
Thank you. Okay. Ronnie, you had your hand up? Did you want to actually say anything as well? Please take yourself off mute if you did. Okay. Next, we'll go to Robin.
Hi, how are you? It's Barry.
Yeah.
How are you?
Good.
Thank you very much for your presentation tonight. It's been very well. Yeah, wonderful. The question for the
Thank you.
night is, do different hive types benefit the cleaning effect of the bees?
Sorry, say that again.
Do different hive types, Wairau, Kenyan, Nadiring rather than supering, does this animal husbandry help the bees cope with varroa?
Yes.
Wonderful.
Yeah. The Langstroth, the Dadants, the vertical lift hives are kind of encouraging us to go in and dig and pull and, we call it fiddling.
Yep.
There's a lot of fiddling, and fiddling is not good for bees.
Yep.
My friend Custis Apium, he builds circular hives. The circular hives are good. The top bar hives are pretty good. I have a couple top bars in my hives, with my hives. Ordinary, if you want to, one of the things is, and I didn't get to say it because of the time, but insulation is very important. Thick-walled hives are much better than ordinary, and Warri has definitely shown a good response to helping with the natural setup of bees, because you don't disturb the brood. That's the key.
Thank you very much. To answer the other lady's question about opening every 10 days, our law in New South Wales says every 16 weeks. So it doesn't have to be every 10 days. The law specifically says every 16 weeks or 4 times a year. So that was only, the 10 days is only bluster by the DPI, not law.
Right.
Thank you for that. Okay. Next we have Ronnie, you've raised your hand. Did you want to say something quickly?
Yes, thank you. Sorry, couldn't find the mute button.
That's okay.
Paul, thank you. Fantastic talk. We run in Tasmania down here. A lot of people are using the Russian Lazutin hive, which is getting even better results than the Wairau hive around not disturbing the bees and keeping a really good environment for them. I've got a little side question here. You mentioned sugar feeding, which I absolutely abhor. I think it's one of the worst things we can do, because it just totally trashes the acidity of a colony
Right.
and the hive environment. Have you seen any research about acidity levels inside a colony and the impact they might have on varroa?
I did see something concerning using the acids like oxalic acid and formic acid, but I could find it for you if you want. The whole thing is that when we put anything into the hive and we change the chemistry in the air, in the environment, we affect the bee's natural situation. But as far as actually saying that, and I hope I'm hearing right, actually say that acidic situations help the bees. Is that what you said?
No, I'm talking about their natural acidity level, which usually sits at about 3.8 or 4.2 in a colony, and that if that's maintained at their natural acidity level, whether that helps varroa.
I would imagine yes. When I say helps them, helps get rid of them.
Yeah.
Is that what you're asking? If it helps get rid of varroa?
Yes,
or
yes.
control varroa? Well, I don't have an exact answer for that, but I do say that if the hive is in balance, the pheromones, the acidity, the food situation, the bees have time, then you can take care of varroa, you know?
Yeah. Thank you.
Okay.
And I'll support what you said. I do a lot of abandoned hives. I have never seen an abandoned hive with any disease in it whatsoever. I
Fantastic.
fully support. You leave them alone. Don't disturb them.
Absolutely.
Hey,
Thank
you're
you.
from Tasmania. You're from Tasmania.
Yes.
I think you're in a situation in Tasmania where you guys, if you really pushed for it, could actually have a situation like Cuba. If everybody in Tasmania just said, "Hey, we don't want to treat, and you've got a high level of Apis mellifera. You've got really high levels of the dark bee. Are you seeing presence of dark bees? Like, that's my question.
Dark bees are all across Tasmania. They are in really high proportions. We actually have a sanctuary of about 250 hives of dark bees, which have very high mellifera genetics. But we are also, Eric's online tonight. He's going to get a swarm trap of dark bees to fill his hive. Quite a few of us are going to be looking at getting dark bees into our makeup.
I think it's fantastic.
Yeah.
I think it's fantastic. You have it there. You have the key right there in Tasmania.
And it's an interesting subject. Sorry, we're taking a bit of time. When we cross Apis mellifera deliberately with other European subspecies, the bees can get incredibly cranky.
Yeah.
And each generation gets crankier and crankier. However, when they are naturally absorbing the mellifera genetics, we are suspecting that they're not getting as cranky, that they're actually sorting that process out themselves. So it'll be interesting to watch.
Fantastic.
Very interesting. Keep in touch.
Yeah,
Keep in
we'll
touch.
do.
I'm really interested, you know. You could get on my mailing list I have it here, www.regenerativebeekeeping.com.
Fantastic.
And
Thank you.
keep in touch. I think you have a very interesting situation that I am personally interested in.
Cool.
Thank you. Thank you. Okay, so Gypsy, we've spoken to you, so I believe you just haven't taken your hand down yet. Thank you for that, Ronnie. And now we have Dave who wants to ask a question. So please go ahead, Dave.
Yeah, g'day, Shelby. Can you hear me okay?
Yep, we sure can.
Yeah, I just want to clear a couple of things up. Firstly, with regards to Ronnie saying she's never found an abandoned hive with disease, I have found tons of them with AFB in New South Wales. So that's I wouldn't put comfort in hives being abandoned and not have disease in them. They are out there, and they're definitely a source of disease for other people. Just wanted to help Kay out a little bit and respond to Gypsy's comment. There's actually no law requiring inspection 16-week for varroa. There's no control order anymore in New South Wales. Basically, all you have to do in New South Wales at the moment is manage varroa like any other pest and disease, and you have to report if you find a positive finding. So, Kay, if you're going in every 10 days, that's too much. The recommendation at the moment is to check monthly if varroa is in your area or if you have varroa, and that's purely been put out there on the basis, and it's only a recommendation, that we've actually found hives in New South Wales where we've had 0 results from alcohol wash, and 66 days later the hives are dead from varroa. So it's up to you how often you check. I'd suggest 10 days is way too much and way too much work for you. Very happy to reach out and help you with that if you want Shelby to pass on my contacts as well.
Sure. And Dave, do you want to just explain your role?
Yeah, so I'm the New South Wales Extension Engagement Coordinator, so I'm in charge of the varroa development officers in New South Wales, and they're a group of 7 beekeepers in New South Wales, and there is videos in other states as well, which have been brought online to help people learn how to monitor and manage varroa going forward using the whole range of techniques, everything from cultural controls to mechanical means, and then into the organic and then the synthetic pesticides. So, I mean, I've sat here and listened to a lot of stuff tonight, and Paul, all good information. I really, really fear that if we just leave our hives alone and wait for things to happen, we're probably not going to have any hives left. If I was to leave all, from what I've seen, if we were to leave all our hives, you know, in an apiary of 100, pretty sure you'll have 100 dead from what we've seen. So I'm really concerned.
Yeah, but can I say something? I think you're misunderstanding what I said, because I particularly said, you know, like you just take a small part of your apiary, like a 10% or whatever, 1 or 2 hives, and everybody form like a group somewhere. You know, I believe Australia has a lot of feral colonies. I know they're going to take a hit, but they're going to come back the first. And the other thing, as I said, is that you continue to treat your existing apiary until such time as you can breed some sort of resistant genetics. I mean, can I ask you who is doing that in Australia? Who is working on building resistance and checking on that?
There's various breeders in Australia who are looking at varroa sensitive hygiene and testing for it to get it into the genetics. But, you know, if you say take a small amount and put them somewhere else, they are going to die. We are seeing huge levels of reinfestation, massive levels of reinfestation. My fear is that, you know, if we get together in 18 months' time and get all the people on this back together and say, all right, well, if we sort of, and there was a comment in it, what do we do? Do we, you know, a comment made that what do we do? Do we wait till they die? Like if there is resistance out there, my fear is that it's going to be in a really, really small proportion of the population of bees. And we're not going to get through this initial hump if we don't do something. That's and I've spoken about this to you, Shelby, so this is nothing new. I'm not bringing anything new to the table here. I guess I've seen the worst of it so far, so that is what really stresses me. And look, the other thing I was going to say too, Paul, I understand what you say, you know, well, the pesticides and stuff have a negative impact. We've got huge areas of Australia where there's no pesticide use. We've got massive amounts of forest area where there's just feral bees. And so if there is resistance there, that will develop within those colonies, I believe, in the bush where they are away from pesticides and they're not being treated. But the time frame for that is what concerns me. And in the meantime, we've got, you know, hugely pollination dependent food industries, which are basically, you know, if we don't, you know, treat, then, and no one wants to use chemicals. I don't want to use chemicals, but if we don't treat, we've got real food security issues here as well to consider. So look,
that is a very relevant thing. I totally understand. And this is exactly what has happened to everyone who has gone into the treatment thing. I mean, Australia is a massive country, and you cannot just decide the whole of Australia is going to just go treatment free. I completely understand that. But finding the genetics and training people to look for those type of traits is what is really important. Taking a very small amount. I mean, you're talking about those areas. I heard that there was about 123 hives per square kilometer of feral bees. Is that correct? Is that a right thing?
There was a study done by Dr. Ben Oldroyd in the late 90s in northwest Victoria, which found feral hive populations of between 50 and 150 colonies per square kilometre.
Right.
That's like having a commercial load of bees dropped every square kilometre, which the concern we have is that that's like having those dropped every square kilometre across the state and not be managed for varroa. So the reinfestation that we're seeing as they die out is just absolutely off the scale. And look, and in terms of people looking for those, yes, there is. There's already people that are doing, you know, they're doing harbo assays. They're using the OBU pheromone for the bees to pick the bees that detect the unhealthy brood. They are looking for these uncapping traits, and they're trying to get that into breeding programs for sure. That is happening.
You know, Dr. Mart, I have a list of resources here that help me with this presentation. Dr. Dr. Martin in England of the British Beekeeping Association, the BBBK or whatever it is, just talking because the British are now already saying it's absolutely true. We have, everybody's starting to go with the regenerative type of beekeeping, the treatment-free, and they are training people and they're showing people how to look for the traits that are there. Now, I completely agree with you when it comes to, I know it's a dilemma. You can't all go treatment-free. It's too big a situation, but some people can. I like the Tasmanian thing, I'm seeing a lot of the Apis mellifera there, and they are showing resistant traits up here. And it's all over the world. The bees have responded to no treatment in different places. It's worth trying it out. It's worth trying it in different places, like out in the bush there. And I only from movies I can see Australia has some serious bush. Yeah. What do you think?
Australia's massive. We have huge areas of bush. Yeah, it's incredible. I just got a message back there from Kay as well. She's already been in touch with videos, so that's good. Anyway, that's really why I jumped on Shelby, just to try and help Kay out.
Okay. I didn't realize I was looking away.
You're on mute, Shelby.
Sorry, I was just saying thank you for clearing up the confusion around the reporting cycle for the current instructions regarding that. So thank you for that, Dave. Okay. All right. So we've gone over time. I appreciate everybody being on the webinar this evening. Thank you for all your comments. Person A, is there anything that you would like to say as a final comment, because you haven't actually spoken yet and you've been adding some information into the chat? Please feel free to do that if you would like to. Okay. Having said that then, I'd just like to thank Paul and all of our participants today. That was actually fascinating, and I will certainly be rethinking how I actually engage with my bees moving forward. I'll be looking for that rhythm and how I might be more in harmony with my bees. The recording of this event will be sent to attendees in the next couple of days. And as Paul is a B&B Varroa Warrior member and a group expert, if you have any additional questions or comments, you can participate in the chat that is attached to this event within the B&B Varroa Warriors Facebook group. We look forward to meeting up with you again at one of our future webinars or events. And you can check out when they are scheduled by joining our Facebook group and accessing the events tab. So once again, thank you very much for your time this evening, Paul, your time during the day. We really appreciate it, and we will now look at closing this webinar. Thank you, everybody.
Thank you.
Thank you. Anything again?